Hello everyone. Thank you for coming to my presentation today. My name is Probert, so I manage the technical team within CPI Biologics. Today I'll be giving a presentation looking at the past, present and future of mRNA manufacturing. Just before I go into my presentation, I'd just like to start by thanking Thermo Fisher for inviting me to talk to you all today. So in terms of what I'm planning to cover, a brief introduction to CPI, just to give some context and the type of work that we're doing and I'm involved in this type of work. Before we then move to looking at RNA technology, so what the technology is, how it's applied. That then feeds into RNA manufacture, so what's been done historically and what's the current approach to manufacture of RNA-LNPs, and then looking forward, how manufacturing may change as we look to consider the future of manufacturing. With a final look at just a few set of conclusions based on what we've run through. So CPI as an organization is one which helps other organizations to develop, prove and scale up, as well as commercialize their new products and processes. This is across a range of different capabilities, so everything from biotechnology, biotherapeutics, formulation, principal electronics, and so on. I'm from the Biologics business unit, which is where our biotherapeutics capabilities and expertise is based. And this takes place within the UK. So most of our sites are based in the northeast of England, so just south of the Scottish border, effectively, between Newcastle and York. But we also have a small molecule manufacturing innovation centre based in Glasgow, just north of the border, just west of Edinburgh. In terms of the work that we do, so that support of other organisations, so we see ourselves as occupying the space between research and innovation and then commercialisation. So helping organisations with their new products and processes, helping them navigate that landscape to generate the data they need to secure onward investment, as well as turn their existing processes into ones which are suitable for commercialisation in the context of biologics to processes which are ultimately suitable for GMP manufacture. And just by way, I guess, context of the organisation, so the business has been open for almost 20 years. Large number of projects completed, now a large number of collaborations, so we work with everyone from universities, SMEs, small, large biotechnology companies, as well as large pharma. In terms of staff, this slide is a little out of date now actually, so we have probably close to 600 staff within the organisation, with a turnover number just to indicate kind of a flavour of the type of size of organisation. In terms then of biologics, which is obviously more relevant in terms of we talk about RNA. So this is a centre that I work at and manage a team in. So this is a relatively new facility, so it was opened in 2015 with work starting proper kind of towards the end of 2016, 2017. We work across BSL-1, BSL-2 organisms, primarily to ISO 9001 standards for our scale of manufacturing process development. But we do have GMP facilities coming online in the last part of this year, which I'll say a little bit about later on in the presentation. In terms of generally the kind of work that we do, so we work across everything from cell-free microbial and mammalian expression systems used for production of everything from antibodies, recombinant proteins, viral vectors, as well as things such as plasmid DNA and obviously of relevance today, mRNA. In terms of our capabilities in the space, so we have a kind of a general process offering for biologic molecules, but mRNA and LNP specifically. So we have expertise in support of architecture design, of RNA synthesis, and scale up to relevant manufacturing scale. We have expertise in purification, so again, small scale, high throughput screening and scale up. We have the ability to support encapsulation, so most of the work today has been working with a standard 4-lipid system. And we have different encapsulation systems, both T-piece approaches as well as microfluidic mixers. And again, with the ability to scale up to that suitable for manufacturing. Most of our work, commercial basis is preclinical supply currently, so production of materials support tox studies. But with the GMP facility coming online later on this year, we'll have the ability to support production of material for clinical applications. All this work is supported by our analytical characterization expertise, so we've put a lot of time and effort into this to develop not only kind of the requisite assays in terms of identity, impurities, etc., but also things such as, say, structure characterization, which we think is something which is important for understanding, particularly mRNA and siRNAs in processing, and something I think which helps us develop optimal processes. So that's introduction to CPI. So now move on to the thinking a bit more about technology. So when we talk about RNA technology, what is it we're actually talking about? So there are a number of approved nucleic acid therapeutics on the market, approximately 20 that have been approved across by the FDA or EMEA. The majority of these products are non-viral mediated. So we have some products which are viral mediating, such as say AAV products, which have a viral vector, but non-viral mediated actually, perhaps surprisingly for some, are perhaps actually the larger class of approved products. So of these products, as you can see on the left-hand side, we have some, essentially, the majority of these are oligo products, so they're relatively small nucleotides, so whether they're antisense oligonucleotides or small interfering RNAs. These products function through modifying levels of existing pre-mRNA or mature mRNA in order to regulate expression of a given protein. It's worth noting that things such as siRNA, for example, can be delivered through lipid nanoparticles. Lipid nanoparticles are not the sole domain of mRNA, but they do have multiple uses. Where we're talking, the bulk source there is thinking about mRNA. So mRNA, obviously, popular topic in the news due to the COVID vaccine. So mRNA is distinct from some of these oligonucleotides because with an mRNA, we're introducing a mRNA directly into the body that is then going to be used to create expression of a protein. With vaccine products, that's used to create an antigen in potential future therapeutics. It will be used for production of a therapeutic protein. And then with what we've seen with the products to date, so applications to COVID vaccines, but also I think the real potential that people recognize technology has, is it has the real ability, it may be disruptive in this space to mRNAs as the basis for treatment of diseases that are otherwise difficult to treat as vaccines for diseases which are otherwise proven difficult to vaccinate against. So this then just shows a kind of selection in terms of what the breadth of type of products that are being investigated. So currently there are the only approved products, mRNA products, are COVID vaccines. There's a range of other COVID vaccines beyond those which are approved, which companies are looking at taking through clinical trial. But particularly beyond just COVID vaccines, think about other types of vaccines, but also therapeutics. There are a range of other products in trial, so everything from metabolic disorders to cancer treatments, as well as for vaccination against some of the more difficult to vaccinate infectious diseases, which I guess we've seen on the right. I think what's exciting about mRNA technology is it's quite a flexible platform for reasons I'll come to in a moment. However, it's worth noting at this point that in terms of the overall potential of the platform, at the moment the only realised potential is for, say, these COVID vaccines. But in terms of the broad application of mRNA, there's a lot of really exciting applications that may have, but this still remains unrealised potential in terms of the ability to get products approved by, say, FDA, EMEA for more widespread application. So in terms of technology itself, when we talk about mRNA, what are we talking about? So the two main approaches used to date have been kind of conventional mRNA, whether that's with or without modified nucleotides, which I'll come to in a moment, as well as self-amplifying RNA platforms. So the current approved products on the market are both mRNAs, both with modified nucleotides, but actually there's a lot of interest in self-amplifying RNA in a number of different organizations. Irrespective of which format it is specifically, every mRNA is going to have similar type of structures. It's going to have a cap with the 5' ends, which is required for function. We're going to have some untranslated regions, which effectively they exist in the mRNA sequence, but they're not translated within the cell to make a protein. So they're there for stability and enhancement of translation. We have a gene of interest, which encodes for the protein or the polypeptide that we're trying to express, which is obviously the basis of the vaccine or therapeutic mechanism of action. And we have a poly A tail, which is just a string of adenines, which effectively again is required for expression and stability, particularly. When we think about self-amplifying mRNA, broadly speaking, that's quite similar to the structure, but what we have are these additional non-structural proteins that sit upstream of the gene of interest. These are derived from from viruses even. And what these do is they basically encode a replicator, then replicate the entire sequence. So in principle, we can inject a small amount of self-amplifying mRNA into a cell, and it will effectively amplify itself. So we can therefore lead to either the same or greater level of expression than we might have from a more conventional non-amplifying mRNA. Importantly, though, so regardless of what format we're using, we need to then encapsulate that mRNA into some kind of vector. So what we're talking about today is lipid nanoparticles, although there are other approaches to encapsulation that are being explored and may well be the overall future of the technology. And the reasons for encapsulation I'll come to in a moment. But effectively, through this, we end up with an mRNA-LNP that we can then use for treatments. So then in terms of then design, so I've mentioned some of these elements. So we have our CAT, we have our untranslated regions, our target protein, our poly-A tail. Without jumping into lots of detail over absolute specifics of some of these, I think overall, irrespective of the product that we are making, whether it's a vaccine or whether it's a therapeutic, effectively what we are encoding with an mRNA is a polypeptide chain, and that is what we have our clinical effect. Now, because there aren't that many, because the mRNA effect is going to consist of different combinations of effectively 4 nucleotides, we can create very different proteins through quite small modifications in the chemical property of the overall mRNA, compared to something like a protein where there's over 20 different amino acids. Those amino acids have very different properties, different charges, different hydrophobicities, and that means when we create a protein that's although it's still composed effectively of the same building blocks, so one protein to another protein, there can be a huge difference in terms of how its kind of chemical properties, so how hydrophobic it is, how the charge that it has. Now that's the basis of how we purify biologic recombinant proteins, but it means it's difficult to maybe have a very established platform process because they're handled very differently. Within the mRNA, although the nucleotides vary and there's different options, there are only, in most cases, 4, and actually they all have quite similar properties. So they all have the same charge, they all have the same kind of level of hydrophobicity. So broadly speaking, one mRNA is quite similar to an mRNA, not identical, and there are some distinctions which I'll come to, but broadly speaking, they're quite similar, which I'll come to the relevance of that slightly later on. Now, one is we do have the nucleoside sequences that may in the future lead to more variability in different mRNAs is the incorporation of modified nucleotides. So mRNA can be synthesized with different nucleosides incorporated, indeed with the current vaccines both making use of an N1-methylpseudouridine. The identification, I guess, maybe discover the effects of this being one of the reasons it's been given in terms of why the vaccines or why mRNA technology has been able to proceed to where it is now, where we have approved products on the market. So N1-methylpseudouridine reduces immunogenicity, which is a real challenge in terms of mRNA technology, but also gives high level of protein expression, which again means we don't have to give as much, which again then ties into the potential immunogenicity of the product. So for current applications, N1-methyl is, I guess one of the main ones people are interested in. There's other things such as, say, methyl-5-cytidine that people are interested in. There's still a bit of discussion over the incorporation of modified nucleotides, although I think generally most companies, particularly from most kind of standard mRNAs, are looking to incorporate them, given their upsides. But there is still further research to be done with these, but also generally in terms of nucleotides in the market, there's a huge range of modified forms that can be purchased. The effects of many of these is yet to be defined, and in some cases, the research on some of these modified nucleotides has been done before some of the advances that have been made since then. So therefore, what was known about them then may not be relevant now in terms of other changes that have been made. So I think what we see at the moment is there's kind of a reasonably standard approach that people have tried, but I think in the future we'll like to see the incorporation of other types of nucleosides that we may not even know about at this stage, that may have a significant impact on the function of the mRNA, but also in terms of how it actually processes and handles. The only thing related to that is, with that change, we may also see a need to change some of the polymerases, so the enzymes that ultimately create the mRNA, because some of those enzymes have more or less ability to incorporate different modified forms, and therefore we want to go into more exotic nucleotides. We may need to look at changing polymerases, and the relevance of this again I'll come to in terms of, because it relates to supply chain, which is something which I believe is important in terms of mRNA production. So in terms then of the encapsulation, so I mentioned earlier on that there's a need for mRNA siRNA, regardless of the format, they need to be encapsulated. That's because RNA is fundamentally unstable. It's designed, or its role within cells is to be the message between the DNA and creation of a protein. So it's not necessarily designed to hang around for a long period of time. DNA ultimately is the hairful material of the cell. So therefore, if we want to get stabilize mRNA in the body, we need to encapsulate it, but also mRNA has a strong charge and therefore isn't going to naturally cross the plasma membrane of cells. So we need to also help it in getting across the membrane so we can then release it into cytoplasm and it can be translated. So the vector system needs to protect it from nucleases in the body. It needs to evade systemic filtration so we don't introduce our therapeutic. It's immediately filtered out of the blood and therefore can't reach its target tissue. It then helps it reach target tissue, internalize it and release it so it can be translated. So there are a range of different options kind of available with kind of relative different pros and cons. And again, I think in the future we'd like to see some, I think the encapsulation technology is really important in terms of the composition of lipids and how we target our mRNA or siRNA to our given target tissue. In terms of where things are now, there's probably a reasonable standard approach in terms of using LNPs as the basis for mRNA or siRNAs, although again not ubiquitous and may change in the future. And typically these are based around say like a 4-lipid system. So different lipids playing different roles to ultimately create this lipid nanoparticle. So I'm not going to go into chemical structures, but effectively we have things such as cholesterol and DSBC, which ultimately required formation of the LNP. We have stealth lipids, so a pegylated lipid, which improves the half-life of the nanoparticle. And we have the ionizable lipid, which is really important because it enables us to both encapsulate the lipid in a fairly effective manner, but also to release the mRNA into the cell once it's been endocytosed into the cell. So it's a significant, the ionizable lipid was really important in terms of, again, progressing the overall technology because the ionizable lipid is much more difficult to make, to encapsulate the lipid, but also to release it in the cell. So having gone through a very quick whiz through some of the basic technology, it's then thinking about how that then applies and how we actually manufacture RNA. So for us, I guess to provide some context on the work that we've done. So we, within CPI, did some work on some self-expression, which related to doing some kind of mRNA IVT work. We were involved, though, in 2020 with the development of the siRNA from Imperial College London, supported by Vaccine Task Force, to take that vaccine, to bring it in, to do some optimization and scale up, to ultimately produce a manufacturing process for an siRNA-LNP product. And that for us gives it really, I guess, the opportunity to develop a lot of expertise very quickly, which is subsequently built on as well. But again, it's given us exposure to kind of a wide range of different formats of initially siRNA, but now we do a lot of work with mRNA as well. So in terms then, I guess, of a standard manufacturing approach, so this then just shows kind of the approach, kind of the standard set unit operations you will take in terms of manufacture of mRNA. So typically we're starting off with, say we're starting with supercoiled DNA, so supercoiled plasmid DNA. We initially go through a linearization step to create a linear strand that is then used as the starting material for the in-vitro transcription reaction. So the IVT is where we actually take our DNA and with addition of polymerase, we then create a mRNA or an siRNA. Post that reaction, we add a DNA to remove the DNA template out, so it's easy to remove, and we then go into purification. So some TFS, some chromatography, different processes can have some permeation to this. Some actually don't use chromatography at all. Some might have additional chromatography step, but again, it follows a similar process flow to say other types of biologic products. But obviously, looking at removal space, very quite different impurities. Once we have our purified mRNA, post this second TFS step, we then go to encapsulation, where we take our products, we take our lipids, we use them, we combine them to encapsulate. We then do some additional filtration and then final fill. Now, based on the similarity between different mRNAs that I mentioned earlier on in the presentation, the manufacturing flow doesn't necessarily need to deviate hugely for different mRNAs. So we may be using similar reaction compositions, although potentially using different modified nucleotides. But broadly speaking, from a purification process onwards, we know because the IVT process is defined, we know what our impurity profile is, and therefore we'll typically try to remove the same things regardless of what the mRNA or siRNA is that we're working with. What is important though is that there's still, within size of products, we can get different, say, secondary structures in terms of how the mRNA interacts with itself, but also the size has an impact, or can have an impact depending on the process we're following. And that stuff we need to take into account in terms of how we change the process for this particular type of product that we're running through. I think what we've seen consistently, and given the biochemical nature of the process, is that high quality starting material is absolutely fundamental, in that a low quality material will give you a low quality product, and it's difficult to rescue a low quality product if your purification process is based around the removal of impurities. And I'll talk a little bit more about the importance of raw materials in a moment. In terms of analytics supporting the process, so there's a relatively similar set in terms of the parameters we're trying to understand from, say, mRNA platforms as we are from, say, a recombinant protein and mAb platform. So still interested in things like identity, impurities, concentration. But however, the methods we're using for these are distinct. So there's a lot of use of, say, electrophoresis, reverse phase, HPLC, but effectively looking for the same type of thing. The main distinction beyond different types of assays is particularly within the impurities, where for a conventional cell-based process, we have a soup effectively of biomolecules, so protein, DNA, RNA, that we're trying to, say, pull our particular protein out of. And when we're measuring impurities, we're measuring a lot of host cell impurities. Within the context of a mRNA process, we know what materials we put in because they have a spec sheet that goes with them, and therefore we know what it is we're trying to remove. So our impurity analysis is much more targeted because we know exactly what it is we've added and therefore what we're trying to measure. And that's quite a big distinction in terms of not necessarily the type of assay that we run, but the type of quality standard we can expect from one of these processes. The LNP aspects are effectively our drug product. So although we often talk about the formulation step, but obviously it's really critical for the actual function of the product. So again, some of the things we're looking to measure here, things such as encapsulation efficiencies, as well as LNP size and polydispersity index, so essentially how homogeneous our distribution is. But again, there's still some work to be done in terms of defining what's required from a, say, regulators over specification, but I think generally there's a movement towards agreeing a fairly standard set of analytics. So when we think about then manufacturing, actually specifics what goes into some of these processes. So I spoke about this process being a biochemical process, and this is the heart of the process in terms of where we actually make the mRNA. So this is the IVT, the in-vitro transcription. We take our DNA template that's been linearized. We then add a few enzymes, a few substrate in the form of nucleotides, and we basically mix these together. We incubate them at temperature, typically about 37 degrees, for a few hours. And within that period of time, we end up generating gram per liter scale quantities of material, which we compare to, say, a cell-based process. Even a microbial process is going to take a day or two. A mammalian process will take a couple of weeks. So it is exceptionally quick overall, and also is really supported by having access to the raw materials early on. And again, when we talk about impurity profile and purification, because we're only adding maybe 10 to 15 different components, when it comes to the purification, we know exactly what we've added effectively, and therefore we have a very defined impurity profile, which therefore gives us the ability to actually, rather than, as we might a cell-based process, trying to pull our product out of kind of effectively the soup, in this process, actually, we know what we're trying to move, so actually we can always target the impurities removal and keep our product separate. So it's interesting in terms of how we go about defining processes, but also because the impurity profile is typically very similar for different types of mRNA, we don't necessarily need to hugely change our purification process. It's more about aligning the purification process to the specification we're trying to achieve. And again, because the mRNA is very distinct compared to the other like enzymes or substrates in the reaction, so mRNA is big, it's got a strong charge. The DNA template is also quite big, but that gets digested within the reaction step, but that's effectively gone. It's much smaller, so therefore within these kind of processes, there's basically a single CFF step can remove the majority of your impurities, which is very unusual because typically TFF is more of an aid in many steps. We're used more later on in the process as a purification step. Then it can be difficult to move with double-stranded RNA. I'm not going to dwell on this, but it is an impurity that comes out the IVT that is can be quite similar to the RNA in terms of, say, charge and size, which is more difficult to remove. And there are different ways, though, of removing it that have been published and people are thinking about incorporating into their processes. So in terms of a schematic view then, so again, this is just, I guess, the purpose of unit operations, where we have our first TFF step, which actually does a huge amount of the removal of impurities. We have a chromatography step where we're removing, we can use this, well, to target different types of impurities or maybe to try and enrich high quality mRNA, before we have a second TFF step to just remove further any further impurities, but really to do a buffer exchange of concentration leading to encapsulation. When we talk about encapsulation, the typical process is really we take our mRNA within the aqueous phase, we take our LNPs within typically an ethanol phase, we mix them together, and basically by virtue of the change in solubility, as well as change in pH, we effectively result in an encapsulation of our mRNA, and that's really enabled by the ionizable lipid, which interacts with nucleic acids and is incorporated into the LNP. So this is really a mixing challenge as much as anything else, and that's why different technologies that we see, whether it's a TPs mixer here or a microfluidic mixer, is trying to give really good mixing to create a consistent quality product out at the other end. So when we think about manufacturing then, so I think I highlighted that there's a very short process to actually make the mRNA, and that's really enabled by the supply chain. So the manufacturing process is relatively straightforward, and it can be done on a small footprint. That's not to say there's not a huge amount of work required to get to the point where you can actually make that mRNA, because you're working a number of enzymes. Those enzymes all come from typically an E. coli-based fermentation, in some cases from a yeast-based process. So they're produced, they're purified to really high quality, and that's what we then take into our IVT. So supply chain is particularly important within this kind of process. In a cell-based process, we're starting with cheaper or less complex raw materials, and we're letting the cell do the work to then create our high-value product. And even just being things like beyond the enzymes, things like the nucleotides, the cap can be quite specialist. The template DNA is a real challenge because it's specific to a given product. And because the template DNA is really important for overall product quality, it needs to be manufactured at a high grade, and that can be a real challenge in terms of getting hold of that to the right grade on a reasonable time frame to not hold up manufacture. So because these materials are very specialist, they're not that easy to make, they have a high cost. That high cost feeds into the mRNA-LNP. However, importantly, from where we were maybe a few years ago, actually, although some of these things are already being manufactured by suppliers, the supply chain has really tried to keep pace with advancement technology, and therefore actually it's easy to get hold of some of the materials. The costs have gone down, and that's really important in terms of supporting technology, reducing costs, which ultimately has the benefit of improving access to these types of molecules or products to the market. Just that perhaps complication to some of this is that the supply chain has moved along, and there's been a lot of advances made within the mRNA-LNP technology generally. But as you might expect, a lot of the IP is very valuable, and because it's quite a new field, there were, I guess, some companies historically that have obviously done a huge amount of work. Actually, they've, as a consequence, hold quite a large number of patents. That can make it quite difficult in terms of entering the market, because some of the technologies you want to get access to or use that are currently really required for functional products are patented. So therefore, they need in some cases to secure licenses. I think that's a consideration in terms of development as well as cost of goods. In terms of the overall landscape and direction of the landscape, so currently it can be challenging. There's lots of patents, and it does make it difficult to enter the market. But in terms of as more companies want to gain access to technology, that hopefully will lead to licenses becoming more available. But also, given how quick the technology is developing, I think we'd like to see that some patents may become less relevant, because actually people are doing new pieces of work, and actually therefore what was patented and very valuable maybe less valuable than it was. There's something to watch, something to be aware of in terms of manufacturing. So in terms of, I guess, past and present, so I've not specifically stipulated in terms of evidence between past and present in a huge amount of detail. I think what I'd summarise, I think, from where things are to where things are now, is that broadly speaking, what was done maybe historically a few years ago is still pretty much how we may go about manufacturing now. There has obviously been a scale up of some of those approaches, but in some cases we're still using similar types of technology. For example, like reverse phase-based purification, which is say done at academic labs, but can be scaled up to a point. We're primarily working, say, batch-based processing as something which is fit for purpose and does make suitable quality mRNA. I think generally, in terms of why we're not seeing huge changes in manufacturing necessarily, is that the technology is really matured very quickly or come to market very quickly, and there's a need as a consequence of the pandemic to move very quickly, and therefore, if the process performs and we know it's effective, then there's no necessary need to reinvent the wheel at this stage. But in terms of generation 2, things are more likely to change. I think what has changed, though, that is maybe not something that is seen directly within manufacturing. The supply chain is really advanced. It's really it's a lot easier to get hold of high quality materials, which actually really supports running consistent processes and generating high quality product. There are some limitations around patents and IP, but RNA, the whole field is developing really quickly. Not to say that with development we won't see new patents things coming that have to be taken into account, but the breadth of some of these patents is likely to be less expansive, given that there's a better understanding now of the technology and the potential value of it. So then if we then start thinking about the future of RNA technology, so I think overall, as I mentioned a moment ago, that the scale production of RNA-LNPs is really quite a recent phenomenon. Historically, they've obviously, there's been clinical trials in these technologies for years, but clinical scale is a much smaller scale than commercial supply. So that's quite new, and I guess the interest in manufacturing is really developed significantly as a consequence of, I guess, general interest in technology. The field generally, though, is still advancing rapidly, so there's potential new RNA formats. So that's an example of incorporation of different nucleotides, but also, say, not just a linear mRNA, but also, say, a circular RNA that there's some developments in, as well as developments of, say, new encapsulation technologies. At the moment, the only approved products are vaccine-based, and there's obviously a lot of interest in therapeutic products, but the specification for these is still not really defined, because there's not a one on the market we can point to and say, well, that's what defines a good therapeutic product. So in terms of where we are now, given that how quickly things are moving, say from my perspective, I think as an organisation which works with different customers and different types of products, there's a need to ensure the level of flexibility of manufacturing to ensure that we can support the products now, but also the view that we can support products that may come through in the future in order to achieve a high quality product that is going to be fit for purpose. Given that we don't know exactly what point we're going to get to in terms of is there a standard process everyone's going to end up running, or actually is there always going to be a diversity in terms of manufacturing approaches. On top of that, so I've mentioned, I guess, supply chain and the cost. So there is a high cost of goods. The raw materials are expensive, but they're also quite specialist, and they are difficult to make. That increases the cost of goods. It limits the work that can be done at representative scale, which does impede process development. But also, some of these raw materials are quite hard to get hold of. So at the moment, things like plasmid DNA was always quite hard to get hold of before because of its use in other gene therapy products, but it remains hard to get hold of, and it can really delay progression of work. So if we can improve the use of the raw materials we do have, that's obviously beneficial. But also, given the rate of movement of technology, we like to see things like new enzymes come in, more new nucleotides come in. Those aren't going to be available in bulk necessarily if they're very innovative or very new. So therefore, again, the more we can get out of existing, or the more we get out of our raw material inputs, the better, ultimately, as well as obviously reducing the cost of goods. So in terms of, I guess, flexible manufacturing, so I've highlighted before in terms of there's a reasonably consistent manufacturing processes that can be applied to RNA formats, obviously within reason. There can be variability in terms of the input raw materials, but different steps are broadly the same. The process themselves stuff broadly scales linearly from small to large scale, and the recognised benefits of mRNA is having currently what believed will need to be a low mRNA dosage requirement for, say, therapeutic applications, say, vaccines is pretty low, as well as high yields that mean we can manufacture in small footprints. It also has, intrinsically has a modular construction in the sense that we essentially have a payload, the mRNA or siRNA, and we have our vector which will transport it. And depending on what we're trying to express, but also where we're trying to get it to, in principle, is the view that we can mix and match our payload and lipids to create our therapeutic of choice. So there's already effectively through all this some intrinsic flexibility in mRNA, but is there like a one-size-fits-all process? So what we see perhaps is that maybe there's not an absolutely fixed platform process that was exactly the same steps, exactly the same, say, chromatography steps, exactly the same IVT that we use a one-size-fits-all. For us as an organisation working with customers, we already see that isn't necessarily the case. So we instead see this as maybe more of an mRNA toolbox, where we have effectively, as you see on the top here, our different unit operations, so our template, purification, analysis, etc. But within that, there may be different things that we need to run through. So, for example, we may have a different type of product, so whether it's got different cap on, a different type of modified nucleotide, it does the IVT run across a one-step or a two-step, how are we purifying it, etc. Again, analytics broadly will remain similar across products, but in terms of encapsulation, are we encapsulating the LNP into a polyplex? So in reality, the specification of these different types of processes will vary, but there's also different considerations that there may be an optimal process in terms of, say, the IVT, but then due to licensing restrictions or patent limitations, and all of it has to go down a certain route, and therefore we need to be able to support that, and therefore it can't be a entirely platform process by virtue of that. So really what we see is, there's all these different types of operations. There's effectively permutations of that we need to understand and be able to take into account. And I think what's really important is then be able to understand how, say, a different IVT relates to different type of purification and what that final specification will be. So the process needs to fit the product as well as the specification. And if we understand how these different things relate to each other, then we should be able to mix and match different parts of the process to ultimately create the product that a customer is looking for. And importantly for us, we see these processes should be agnostic. So if we understand, broadly speaking, the window of operation we have, so what kind of size of mRNAs or what we need to change for a given product within the context of the process, then we don't need to, the process is run according to the specification required rather than necessarily run according to the particular type of mRNA that we are running. So again, a toolbox should give us in principle a relatively flexible approach in terms of saying what's on the market now and to an extent what may come in the future. And this is some stuff we've been exploring through, I guess, subsequent work. So this is work we're doing with people at College London, the UBC, to again look at RNA manufacture, to characterize it, and look at how we can mix and match different parts of the process to try to understand what is manufacturing, but also with a view to try and identify things like CPPs as well as CQAs from the process. I think in terms of other process improvements within maybe a batch phase we'd like to see. So within, say, IVT reactions as a batch process, there's a limit in terms of what we might overall change. The main change within IVT is going to be the raw material input. And at the moment, there's a lot of interest within the purification and the encapsulation part of the process. So for purification, some things like reverse phase based purification use large amounts of acetonitrile. It's not that amenable to scale up. And that can be a limiting step in terms of how we scale up processes. So we're looking at how we remove that. Here's oligo-DT affinity purification is currently the only affinity-based method for mRNA production, which does work, not necessarily completely platformable, because it depends on the length of maybe the poly tail, and also has relatively low binding capacities, but it does essentially enable you to capture mRNA and wash your impurities away. The double-stranded RNA removal, I mentioned a few slides back. This is a purity that's known to be important, although the overall importance bit and specification that's important to achieve is still probably to be defined. But again, there's particular ways of removing that may be distinct. And that's something we may need to incorporate into our process to create a therapeutic that has the level of efficacy that we need. And beyond all these kind of things that are being looked at now, there's also all sorts of other types of purification methods that may come through. But again, like expect to have relative pros and cons, but I guess the question is, will we see the market converging on basically a single purification approach in the same way that say MABs pretty much universally use protein A as a method of purification. In encapsulation, as a range of technology explored, so some of this is with already existing patents and that there's not that many systems on the market that are available, but also again to make further improvements to scale and consistency. This is particularly looking at different types of mixing, so whether it's a ring-based mixer, membrane, or other types of novel microfluidic approaches. And again, in both these purification and encapsulations, unlikely we're going to see a significant change in terms of the overall process flow, but it will change aspects of the process and may change how our future manufacturing processes look. The other considerations beyond the some of the operations, also just that supply chain and the input materials. So it's likely that we're going to see improvements to mRNA-based products, particularly driven through differences in the raw materials as a consequence of how we design the product and what we incorporate within that. So different modified nucleotides I mentioned, these are, I think very likely we're going to see an increased range of more exotic but more unusual nucleotides incorporated that we haven't typically seen. That will require different polymerases and also incorporate those whilst still giving suitable yield. There's also some interest in using different, say, polymerases at different temperatures, for example, to reduce the impurity profile out of the IVT. Beyond that, we'd like to see other enzymes for capping, for example, synthetic DNA templates and alternative plasmid DNA, as well as, I think, different lipids, particularly for therapeutic applications for longer half-life and more specific targeting of your product to the tissue of tissue for the desired mechanism of action. And again, while some of these raw materials may change, most of these go in at the IVT step or the encapsulation step, but actually the workflow is again unlikely to change, but it will change the supply chain, how processes are organized, but actually we still expect to follow broadly similar workflow. So I think in terms of getting to an agile or flexible manufacturing workflow, I think it's really about understanding the different unit operations that we might need and how those affect specification, with some of the follow-on changes that may come through, or maybe things that basically we can kind of feed into that without having to make any necessarily massive overall changes to the process. And that's based on a kind of a batch type approach. In terms of thinking about how we move from a batch to perhaps a more intensified process, so we recognize that mRNA is a high cost of goods. The raw materials can be difficult to get hold of and are expensive. So in other areas with bioprocessing, there's an interest in intensification, so how we can generate more from the same or from a small footprint. So one of the real benefits of mRNA-based production is that it's overall less complex based on a lot of the work's been done further up the supply chain. And therefore, we know what we're adding. There's not that much going on from a chemistry perspective, which therefore gives us the ability to understand as well as control the process. So we can model the reactions, we can understand the utilization of components, how long we can run them for, how we might be able to improve overall yields. That gives us the ability to then look at fed batch production, but also potentially things like continuous production in order to generate more products from the same set of raw materials, or feed in some raw material, but overall generate a much greater quantity of product. And most lots is done within the, say, IVT components where we're doing this reaction. But really needs to be able to keep up with the IVT to ensure we're maintaining a consistent quality profile. On the right here, we just have example schematics of what these kinds of things may look like, where you go from a batch process at the top to then a fed batch process where you start, say, feeding nucleotides, amongst other things, to then drive up your mRNA yields. And purification-wise, I go into detail, but effectively there's a selection of approaches we can take. We can create a continuous IVT that we can then purify our product through. Different pros and cons, but again, because we know what we're trying to remove, we know what the size of them is, etc., and the process isn't going to evolve over time because there's no cells in it, we can model our process and therefore maybe design appropriate purification around that. Once we have purified mRNA, encapsulation itself is kind of by its nature continuous in terms of mixing of lipids and mRNA, so that's again something we can incorporate into the process. I think overall, I think the mRNA-LNP manufacturing is amenable to process intensification because the multiplying process has less variability we might see with, say, cell-based continuous processing. But what this enables us to do is generate more product from the same set of raw materials and potentially manufacture on a smaller scale. So then if we think about conclusions, so what does that all mean in practice? So when I think we talk about process intensification and the other benefits of mRNA is a reasonably consistent manufacturing process of different products, and we don't need to work on a huge scale in order to create a commercially relevant amount of products. So it really lends technology to less centralized production and much more responsive production. So some of the therapeutic molecules or products that may be of interest may not necessarily have a huge range of potential patients out there. Typically, historically, for maybe more conventional biologics, that might make it then difficult to justify making that kind of product, or might make them very expensive because mRNA can be done at relatively small scale, and the process is more platform based, then actually it makes some of these products much more feasible. And if we may see more, personalized healthcare effectively at an appropriate cost enabled by this kind of technology. For CPI, we've established this our Centre of Excellence. So this is where our GMP capability will be based. And again, it's a relatively small footprint for GMP operation, but actually we can generate multi-million doses per batch from this kind of scale of operation that's really only made possible by the RNA-LNP technology. So I think overall then, in terms of conclusions, so I think mRNA has definitely demonstrated efficacy as a vaccine platform, and there's still unrealized potential in terms of its use as a therapeutic. In terms of what's been done to date, what's still currently being done is that the approach to manufacture scaled with the need. So we've needed to move quickly, we have moved quickly, but there still is very much scope for improvement. So if we say what would be done to date the generation 1, what does the generation 2 manufacturing process look like? And the scope for improvement not just in the products, but also the manufacturing process. So the nature of product by nature means it's a minimal platform production. We define a purity profile, and the main approach remains batch. But there is a particular, I suppose, just the last few slides, there's a real potential for process intensification to potentially be able to manufacture on an even smaller scale and really drive down the cost of goods. In terms of other improvements, I think we're likely to see some real meaningful improvements from new technologies. So the process will see some improvements too, but particularly the product as well. So some of these therapeutic products will need really high efficacy and will need to not be too immunogenic. And some of that will only be enabled through not just design of the products, but also things, the nucleotides that are incorporated within them. What we've seen overall is that supply chain is really important for these kind of products by their nature, because we're reliant on having much more complex starting materials that enable us to move really quickly, and enables a lot of the other benefits that we see with technology. What it does mean, though, is the technology advances and we look towards kind of the next generation of product, is that supply chain needs to keep up with that, and it will only be enabled by collaboration with suppliers to ensure that if we identify new technologies or new approaches, that there's some ability to actually buy those materials to the right grade. Because if not, then that ultimately will become rate limiting for new products to market. So that's, I think, plan to present today. So I hope that was informative in terms of capturing, I guess, where manufacturing has come from and where it's actually going. Again, I'd like to finish again by thanking Thermo Fisher for inviting me to speak today. And I believe we're moving into a question and answer session. Thank you.